Hello, my name is George Vandendriessche. I'm a scientist at Biogen, and today I will be co-presenting with Evan Anderson, a solution architect from Tetra Science, about our recent efforts to outline a path towards allotrope adoption through cloud-based data harmonization. Our presentation will specifically highlight a qPCR plate reader case study. So the need to adopt file standards is really driven by Biogen's analytical data strategy. And this strategy expands to how we manage both our analytical instrument systems, such as chromatography instruments from Empower and Agilent, file-based instruments like Beckman-Coulter cell counters, Perkin-Elmer plate readers, or Thermo Fisher plate readers. Currently, all of those systems, the data exists in this siloed state. So information's trapped in servers, it's trapped in PCs, it's trapped in individual files. Those files each come with their own type of vocabulary, and it's really difficult to manage. And so our strategy envisions a pipeline that will seamlessly combine that information with the experimental metadata housed within our lab information systems or lab notebooks. And this pipeline will be automated and transparent and easily traceable to enhance our data integrity. And this pipeline that combines them pushes information into a cloud-based storage system, relying on the use of standardized data models, data standards, and structure. So these standardizations occur in multiple formats. It standardizes the vocabulary that we use to reference our information, not just across instruments of the same type, but across multiple vendors as well. And it enables vendor independence. It allows us to adhere to fair data principles so our information becomes findable, accessible, interoperable, and reusable. And once that information is fair, this gives us new opportunities for data analytics and applications. And so this is automated data reporting from experiment and new insights into historical analysis, automated filing, and the ability to monitor the performance of our instruments and assays. So we can achieve this strategy through key collaborations. One collaboration is with the Allotrope Foundation. So Allotrope provides access to standardized data through the development of instrument ontologies, AFOs, data models, AFMs, and file structures, ADS. And when you combine these three components together, this gives you vendor independence. We can also achieve the automated movement and handling of our data through a collaboration with Tetra Science, a R&D data cloud company. Tetra Science provides automated data pipelines through their Tetra data platform, which Evan will provide more detail about later. This platform provides file conversion services, movement of information into a data lake so it becomes fair compliant, and gives us access to the information through REST APIs and SQL. So the path to allotrope adoption really starts with understanding our current state. And that current state exists of siloed data. And data can be siloed in multiple ways. It can be siloed by vendors, having different vendors providing different instruments and having diverse vocabulary. The second way our information can be siloed is by location. Biogen is a global company, so instruments managed in the North Carolina site, there's those similar instruments existing in our Cambridge site or across the globe. And so there's really this overlying challenge of how do we manage all of those instruments from a remote work situation like we are in now. And finally, you can have multiple instruments from the same vendor that are outputting siloed data. So this means the data is trapped on an instrument PC, in a shared network drive, on a personal drive. And so we need to be able to handle all three of these instances of siloed data by implementing a silo to cloud pipeline to create what we're calling a harmonized state. And our approach to creating this harmonized state is utilizing the Tetra data platform to read our instrument outputs, transition them into the cloud with a intermediate data schema that Evan will highlight later. Once we've achieved this harmonized state, we can then begin exploring the integration of instrument standards. And this process occurs through a file conversion step, so converting into a standardized format like an ADF, archiving those ADF files so that we have access all of the historical information in the same format, and then implementing triple store technologies like Apache Jena, Amazon Neptune, or Halyard so that we can query and access information stored in ADFs. When you combine these three steps together of conversion, archiving, and triple store, you reach a state that we feel is ready for you reach a state that is allotrope adoption. And you can really, so you have from your harmonized state, you can achieve short-term ROI, and from allotrope adoption, you can achieve a long-term ROI. In keeping this in mind, we will apply our short-term goals to a qPCR plate reader instrument. So the overall goal of this case study is to use cloud-based data harmonization through the Tetra data platform to create a harmonized state for qPCR plate readers. The specific instrument I will be discussing today is the Thermo Fisher VS7. This assay supports our in-process development for the detection of host cell protein DNA impurities. The assay we perform about 2,000 tests of the qPCR assay per year, and this instrument is used in multiple locations in the North Carolina and Cambridge sites. Really briefly, I'll just touch upon what a qPCR assay is. So qPCR stands for quantitative polymerase chain reaction. So in your sample, you have a target strand of DNA, and there's a primer that's developed to bind to this strand, and we can quantify the impurity of DNA through a polymerase chain reaction, except the DNA primer also has a fluorescent probe attached. So as the PCR experiment occurs, we get more and more of this fluorescent probe attaching. And through each cycle of the instrument, we get a stronger and stronger fluorescent reading. And at a certain point, there's enough of this fluorescent probe being generated that we can actually quantify the DNA present and determine a cycle threshold. And from that cycle threshold, you can create standard curves that enable you to identify impurities of DNA. And Evan will demonstrate later a dashboard that we've built that automates this experimental analysis for us. The qPCR instrument state within Biogen is that we have multiple instruments. Each instrument generates a CSV or an Excel file containing all of the experimental results. So this includes the instrument parameters for setup, like calibration. It includes the processing data for each cycle, and it includes the output data. These CSV and Excel files can then be pushed into 3 different pipelines. Information can either be stored on an instrument PC, a shared drive, or to a personal drive. So this here presents a very big challenge for us if we need to access our historical information because it's stored in 3 diverse locations. The data from those locations is then pushed through a pipeline into storage through an electronic lab notebook where all of our raw data is being stored, and then an Excel workbook for processing that information. It's important to note that these Excel files where the data processing is occurring are manually archived in ELN as well. And from these Excel files in our ELN, we then have select pieces of information being stored in our LIMS management system for file reporting. And so a quick summary, the qPCR outputs a single file. This information is stored in one of 3 locations on an instrument PC, a shared drive, or a personal PC. We are completing the single file experimental analysis in a manual process using Excel. And the historical data that we are generating is currently unused. And so at this point in the presentation, I would like to I would like to hand it over to Evan Anderson, who will present about how the Tetra data platform has created a harmonized state for our qPCR instrument.
Great, thank you, George. So as George mentioned, I'm Evan. I'm a solutions architect with Tetra Science. And my background is actually in biology research. So I was really excited to work with George on doing this data harmonization. So as George mentioned, I've just kept the workflow that Biogen is interested in implementing for eventually adopting Allotrope, which includes data harmonization. And the way that data harmonization is working here is through the Tetra data platform. In this particular case study, I've highlighted a high-level architecture diagram for how this occurs. So files are acquired at the instrument PC. So files which are produced by the qPCR machine, such as CSVs, Excel, or text files, are saved into a particular directory. And the Tetra file log agent, which is a piece of software that's installed on that PC, watches that directory for new or modified files that fit a specific pattern. When it detects new or modified files, it pushes these data into an experimental data lake hosted on AWS. Once the data hits the data lake, it triggers pipelines in the Tetra data platform that harmonize this data by transforming this raw vendor data into an intermediate interoperable format. This intermediate interoperable format can be consumed for custom analytics and dashboarding via our REST API or through SQL queries. Additionally, production of this harmonization pipeline with Biogen aided in eventual model development with the Allotrope modeling working group. I'm now going to dive a little bit deeper into each of these steps. So as I mentioned, the raw data for Excel or for qPCR is in CSV or XLS. When it hits the data lake, thanks to the Tetra file log agent, it triggers a harmonization pipeline, which transforms this vendor-specific data into an interoperable JSON format, which we at Tetra Science call our intermediate data schema JSON. Once this IDS JSON is in the data lake, it's indexed for search, so you can access it via REST API. It's used to populate AWS Athena SQL tables, so you can access it via SQL. And it's a standardized format which allows for immediate and interoperable usage, either through consumption by data science tools and dashboarding tools, or through pushing to systems such as ELN or LIMS. I'm now going to talk about some of the ways that you can consume this data. So when I worked with George to first implement this solution, we first wanted to try to recapitulate some of those manual analyses that were happening in Excel. And the tool that we turned to do this was interactive Python notebooks. The reason that we did this is because they're flexible tools that are really used by the data science community a lot, where you can do great analysis and also visualization. So if you want to access data which is in our data lake in our data platform using Python notebooks, it's as easy as launching the Python notebook, importing libraries that you need. The Python data science and analysis ecosystem is rich, and there are a lot of tools for doing the kinds of analysis that we want to do. You can make an API call to the Tetra data platform to get your data of interest. Then you can do the data processing that you need to do, as well as the visualization and analysis using Python. Now, this was a great initial tool for recapitulating that Excel analysis and turning it into an automated analysis. But not all scientists are super familiar with coding and programming. So we opted to transition these Python notebooks into I'm not sure what happened there, into a dashboarding environment. Here we go. Yep, okay, so we're back into a dashboarding environment. So the dashboarding environment that we chose was Streamlit. It's a relatively new dashboarding tool that is Python-based, which meant that we could translate pretty much one-to-one from our interactive Python notebooks into this dashboarding environment. And we could also still harness those vibrant Python data science ecosystem. One really cool thing about Streamlit that distinguishes it from other Python-based dashboarding tools is that it's powered by scripting logic. So there's a low development entry barrier for those who are familiar with programming in a notebook environment. The added benefit of going into a dashboarding environment is that we could host this on a server for access by scientists. So we could host it within the Biogen network, and then scientists could access this dashboard via URL. So at this point, I'm going to ask Jamie to turn over control to me so I can demo some of these dashboards that we've developed, which consume data from the Tetra data platform. Wonderful. Thank you, Jamie. Let's see. There we go. And I can see I'm showing my screen. OK, wonderful. So there were two use cases that we were enabling. One was automating that single file analysis that George talked about that currently occurs in Excel. And a second use case, which was previously time prohibited, is doing trending analysis off of all the available data. So first, I wanted to really quickly show you what a Jupyter notebook looks like. Just in case you're not familiar with interactive Python notebooks, this is an environment where you can perform, you can have interspersed text and images along with snippets of code. So in this first notebook that we created for single file qPCR analysis, we have some description of the analysis that we're doing along with the assay that's occurring. We import some of the Python tools that are happening in this cell of code. We perform an API request for a specific file ID inside the data lake, which we can see was successful here. We do some data transformation, and we can do things like create tables of the data for export and create graphs. And once you've implemented the logic here in Python, it's very easy to just standardize it. And I'll just go back up here. The only thing that you're going to be changing is the file ID here. So you can do this on multiple files. However, as I mentioned, if you're not familiar with Python programming, needing to go in and change this code might be a little bit of a stretch. So here's how we translated this into a dashboard. So when a scientist uses this dashboard, what they do is they first select a file. And the way they select a file is by looking for a file within a specific date range. These date ranges are by default populated to the last file which entered the data lake. So that happened to be on September 19th in this case. Right now, we only have a single file we can select from, but if we extend this date range, there are some API calls that go on in Python underneath the hood, and now we have more files to choose from. So for this demo, let's just select plate 2, and we run the application. Now we have 3 main sections that the scientists are interested in that we can explore. One is the raw results table. So if they just want to look at the raw results from the study and do their own external analyses, they can look at this table here. They can sort it however they want to, and they can also download this as a CSV. Secondly, they can observe the suitability of the experiment, such as looking at controls, and then also looking at that standard curve. So that standard curve is what I'm going to show you for the sake of time. Here's the standard curve that's used to calculate the amount of DNA in each well. As you can see here, this is an interactive graph, and it's this standard curve fit that is used to calculate all the DNA. Now, if a scientist decides, based off of some criteria, that, for instance, this standard is a bit of an outlier, since we're working in a dashboard, we can opt to omit this value, and we recalculate the standard curve for calculating the DNA quantities. Lastly, we have the sample analysis section. where scientists, after they've done their tweaking of the standard curve and deciding if the if the suitability is correct, they can export summarized data based off of the samples that are occurring in triplicate as a CSV. So this application was able to standardize and automate that manual analysis that was occurring in Excel in a very custom way. Next, I'm going to demonstrate the power of having all of your data harmonized within a data lake that has where your data is indexed. And that additional one-up that you get is the ability to do things like control charting and trending of an experiment. So in this application, we allow scientists to select a date range that they're interested in of studies that occurred. When I click the Run app, it's going to search for all files that came into the data lake in this date range. And we've identified 193 hits. That happens to be the number of qPCR files that are in the data lake at this moment. And we get 3 sections. One is a table which summarizes the data that scientists wish to trend, which includes the parameters associated with that standard curve, as well as some control values. So they can export this table for external analysis if that's what they want to do. But we've also created some graphs that they're really interested in. One is looking at trending of control values over time. So again, this is an interactive graph. You can export it as an image if you would like. And this graph will be exported or will be updated as new files enter the data lake. Secondly, as I mentioned, scientists are interested in looking at their standard curve parameters over time. And so this application allows them to do just that. And to be able to look at when those parameters deviate from the norm and to see if their new parameters are deviating from the norm. Additionally, since we have all the data here, it enables even more complicated analyses, such as looking at the relationship between all of these parameters. So this is just a scatter matrix looking at all of the different parameters against each other. So at this point, I'm going to go back to the slides. So thanks for letting me share my screen, Jamie. Wonderful. Okay. So just to summarize the demos which I showed you, achieving that harmonized state that Biogen is interested in for later adopting Allotrope has led to these short-term ROIs for scientists. Additionally, production of this data harmonization pipeline with Biogen enabled a deep dive on qPCR terms, which enabled us to kind of create a foundation for ADF model development. And I'll describe how that works here. So when we designed this IDS intermediate data schema with Biogen, we captured 60 terms from the raw data, along with testing out these raw terms in the dashboarding solutions, as well as important input from Biogen's qPCR SME, Devin Bailey. We are able to tune in on 31 of the most important terms for understanding Biogen's qPCR plate reader assay data. We ended up taking these terms in order to propose a model to the Allotrope working group. And the way that this works is, so as I mentioned, we take our prioritized qPCR term list and we take it to the modeling working group. We first brought this to Matthew, who leads that working group, and Ben, who is really, really involved with the ontology. And they worked really hard to bring Allotrope ontology terms to these terms that we identified, as well as to bring standardized descriptions to these terms. After their hard work, George and I brought these terms back for SME review with folks like Devin, who played a really large role in review. And then this could return back to the modeling working group. And through this iteration, we're arriving at a model that we're really happy with and we think captures this qPCR data well. Now we're in the stage of sharing this qPCR first draft model with other vendors to make sure that it really captures qPCR holistically, vendors such as Bio-Rad and Agilent. So once we finalize this tabular ADF model, the Tetra data platform can incorporate this allotrope ontology to create files that incorporate the ontology. And with that, I'm going to hand it off to George for final thoughts.
Thank you, Evan. So just to recap, we have at Biogen, we have this vision of moving all of our data from a siloed state into a harmonized state that can adopt file standards, specifically adopting allotrope standards. And from the harmonized state of our data, we were able to achieve short-term ROI through harmonization with the Tetra data platform. And that enabled custom automated analytics. So where our qPCR assay analysis was manually performed in Excel, that is now automated. So as soon as those results are generated and that file is saved, the Tetra data platform moves it into the data lake. Our Streamlit dashboard performs the entire analysis, and our scientists have their results immediately. We also now have access to all of our historical data to provide new trending insights into our assay performance over time. Using the insights gained from these dashboards, we were then able to begin prioritizing the most important qPCR terms to develop a first draft allotrope tabular model. And this model was a byproduct of a collaboration with the Allotrope Modeling Working Group and an iterative process to align on terminology. And that model is now being reviewed by other vendors to confirm that it has broad application. And when that model is finalized and released, we will then be ready to explore allotrope adoption pipelines for this qPCR instrument. So that means moving into the realm of file conversion, going from our harmonized interoperable data into an ADF file, archiving those ADF files, implementing triple store, and gaining new insights from ontology compatible files. And with that, Evan and I would be happy to field any questions that you have at this time. And we would both like to acknowledge the Biogen and Tetra Science team members who have participated in this project, and then Matthew Fox and Ben Wolford Lim for their help with building the qPCR tabular model. Thank you.